Cells. Cells is known as the fundamental, structural and functional unit of all living organisms. So I'll repeat again. Fundamental, structural, functional unit of all living organisms. And that's what we're going to study in a lot of MCQs will come from this one. An organism consists of one or more cells. There are two types of organisms. One is the unicellular, another one is the multicellular. Multicellular is composed of many cells. Unicellular organisms are capable of independent existence and performing the essential function of life. They perform the essential functions of the life. All plants are composed of different kind of cells. Almost all plants are composed of different kinds of cells. The animal cell had a very thin outer layer known as plasma membrane. Objections to cell theory. The cell theory states that all living organisms are composed of cells and their products. All cells arise from pre-existing cells. Cells showing similarity in chemical composition and metabolic activities. Chemical composition and metabolic activities. Cells are the structural and functional unit of living organisms. Structural and functional unit of the living organisms. The basic structure organization of the typical cells, that is nucleus, the central part and the brain of the cell, which is spherical in shape. It is denser than the surrounding cytoplasm. The nucleus is composed of chromosomes, contains the genetic material. Example, DNA. Cytoplasm, a semi-fluid matrix that occupies the volume of cell. It is mainly composed of water with free-floating molecules. Eukaryotic cells also contain other cell membrane bound distinct structure called cell organelles. Mitochondria, vacuoles, endoplasmic reticulum, Golgi complex. The prokaryotic cells lack all these membranes bound organelles. It is to be noted as ribosomes are not bounded by membranes and are found in all cells. Animal cells contain another non-membrane bound organelles called centrioles, which help in cell division. Outer membrane, the boundary of the cells, which provide protection to the cell and controls the exchange of ions, molecules and other components in and out of the cell. Now look at the size of the cell. The cells exhibit an endless variation in size, lifespan and cellular activities. Example, mycoplasma, smallest cell, or pleuronemo-like organism is only 0.3 micromillimeters in length, that is 0.3 micro-m in length, and the bacteria are approximately 3 to 5 micrometers in size. The ostrich egg, the largest isolated single cell, is about 170 into 135 millimeters. The nerve cell of the human being is the longest cell, having length of 90 to 100 centimeters. Shape of the cell. The cells have also vary in their shape. They may be polygonal, disc-like amoeboid, thread-like cuboid or irregular. All cells can be grouped into two main types. One is prokaryotic cells, another one is the eukaryotic cells. Cell which does not have a nuclear membrane and other membranes bound organelles is called prokaryotic cells. Okay, this is called as prokaryotic cells. Occurrence. Prokaryotic cells are placed in the kingdom Monera, Bacteria are the simplest and the most common type of organism amongst prokaryotes. Size 0.3 to 1.5 micrometers. 0.3 to 1.5 micrometers with some exceptions. Shapes. The four basic shapes are bacillus, that is rod-like, coccus, that is spherical, vibrio, that is comma shape, and spiralium, that is spiral shape of the prokaryotic cell. A bacterial cell is composed of various components as genetic materials: cell envelope, cytoplasm, nucleoids, inclusion bodies, ribosomes, flagella, fibriae, etc. The cell envelope and its modifications. Cell envelope is the outermost covering of the protoplasm of the bacterial cell. It is known to protect the cells from mechanical shock and injuries. Mechanical shocks and injuries. It is the following three layers. Let us see what are the three layers. Glycocalyx, that is mucilage sheath. It is the outer layer made up of macromolecules that gives sticky characters to the cell. Glycocalyx differ in composition and thickness among different bacteria. It could be in the form of loose mucilaginous sheath called slime layer or thick and tough covering called capsules. Function. It helps in resting phagocytes. Next is the cell wall. It is present just below the glycocalyx, made up of peptidoglycan or murin in all eubacteria and cyanobacteria. It is a rigid and solid covering that give shape and strong structural support to the cells. Look at the function of it. So what are the function of these cells? It helps in preventing cell from bursting or collapsing. Okay, it can burst or it cannot collapse. The materials to pass in and out of the cells. Okay, the attack of the pathogens like virus, bacteria, fungi, protozoans, and they provide mechanical support to the cell against the gravity. Look at the plasma membranes. Let's look at the plasma membrane. It is the innermost layer of the cell envelope. It is semi-permeable in nature and is responsible for the interaction of the cell with the outside environment. Interaction of the cell with the outside environment. Next is the function. The exchange of specific material between the cytoplasm and extracellular medium. Selectively permits particular molecules to pass and prevent others. Selectively it allows certain molecules but also prevents certain other molecules. So prevents certain other components through the leakage. The plasma membrane is vital to cellular homeostasis. They are vital to cellular homeostasis. Look at the genetic material. Nucleoids represents the genetic material in case of prokaryotes, that is naked, not enveloped by a nuclear membrane. Not enveloped by the nuclear membrane. Many bacteria contain a small circular DNA known as plasmid other than the chromosomal or genomic DNA. Membranous structures. Prokaryotic cell lack the complex membranes bound organelles. They lack the complex membranes. Some other special membranous structures are found in them. Genetic material. Nucleoids represents the genetic material in case of prokaryotes, that is naked, not enveloped by a nuclear membrane. many bacteria contains a small circular DNA known as plasmid other than the chromosomal of or genomic DNA. Chromosomal of the or genomic DNA. Membranous structure. Prokaryotic cells lack the complex membrane bound organelles. Some other special membranous structure are found in them. Example, mesosomes and chromatophores. Mesosomes. These are formed by extension of the plasma membrane into the cell in the form of vesicles, tubules and lamellae. Mesomers are equal to mitochondria in eukaryotes, as these structures participate in aerobic cellular respiration in prokaryotes. Functions. Mesosomes perform the following functions in the bacterium. So what are the functions mesosomes perform in the bacterium? It helps in respiration, cell secretion. Two things, respiration and cell secretion. Helps in increasing the enzymatic content and surface area of the plasma membrane. Surface area of the plasma membrane. It helps in formation of a cell wall, helps in replication of DNA and distribution of genetic materials to daughter cells during fission. Daughter cells during fission. Chromatophores. They are another membranous structure present in some prokaryotes like cyanobacteria. They are internal membrane system of photosynthetic forms. Which possesses photosynthetic pigments. So it possesses photosynthetic pigments. These pigments are light reflecting. Flagella. Bacteria can be motile or non-motile. Thus, motile bacteria possess one or more thread-like appendages extending from their cell wall called flagella. Bacteria are also classified according to number and the arrangement of flagellum in them. Each flagellum is about 1 to 7 micrometers long, covered by a protein coat. The bacterial flagellum is differentiated into the three parts. One is the filament, the longest portion extending from cell surface to the outside. It is made up of protein called flagellum. Flagellin. Hook, a curved and tubular structure made up of protein subunits. Basal body, the most complex part of flagellum. So all these things are present in the. Next, look at the function of it. They help in locomotion. Example, movement from one place to the another. Pili and fimbriae. The pili are the elongated tubular structure made of special protein called pillin, and fimbria are the small bristles like fibers coming out of the cells. Now look at the function. The pili helps in forming conjugation tube during transfer of genetic materials between donor and recipient cell. The fimbriae help the bacteria to attach to solid surface. Ribosomes and inclusion bodies. Cytoplasm is prokaryotes. Cytoplasm in prokaryotes appears granular due to presence of the following structures. Ribosomes. Like eukaryotes, ribosomes are also found in prokaryotes and serve a common function. That is, acts as a site of protein synthesis. Now look at the ribosomes. Ribosomes are small but are complex both in structure and chemical composition. They are about 15 to 20 nanometer in size. The prokaryotic ribosomes are found in association with the plasma membrane of the cell in the cytoplasmic matrix. The prokaryotic ribosomes are of 70S type. It is of 70S type. Smaller subunits, that is 30S type. Larger subunits, 50S ribosomes, generally occur in helical groups called as polysome or polyribosome. In each polysome, 4 to 8 ribosomes are attached to a single strand of messenger RNA. The ribosomes of a polysome help in the translation of messenger RNA into proteins. Inclusion bodies. They are non-living structures present in the cytoplasm and not bounded by any membrane system. They may either lie free in the cytoplasm or may be covered by 2 to 4 nanometer thick non-protein membranes. Example, gas vacuoles, sulfur granules, etc. Next slide. A cell which has a well-organized nucleus with a nuclear envelope and several membranes-bound organelles is called eukaryotic cells. It is called as eukaryotic cell. All eukaryotic cells are not identical. Except moneurons. Eukaryotic organization is seen in all the protists, plants, fungi and animals. Eukaryotic cell is larger than the prokaryotic cells. Around 10 to 100 micrometer in size. Generalized structure. Eukaryotic cells also possesses a variety of locomotory and cytoskeletal structures. Cell wall is a special membrane being present in plants, fungi and some protists. Plant cells also contain a large vacuole and plastid which are absent in animal cells. While animal cells possesses centrioles which are absent in plant cells. A eukaryotic cell is composed of various cell components. As cell membrane, cell wall, mitochondria, chloroplast, Golgi bodies, ribosomes, centrioles, etc. Okay. So try to remember that. It may come in the multiple choice question. Cell membranes. Cells is covered by a thin elastic transport semi-permeable and regenerative membranes called cell membrane, also called plasma membrane or plasmolemma. It's called as plasmolemma. When the electron microscope was found out in 1950, the detailed structure of the membrane was studied. Very minute structures of the membranes were studied, especially on the human red blood cells. Especially they studied on the human red blood cells, RBC. So look at the structure of the cells. The cell membrane is composed of lipid which forms a bilayer with protein molecules embedded in is at places. Okay. Cell membrane also possesses protein and carbohydrates, and they possess protein and carbohydrates. Lipids. The lipid molecules are amphipathic in nature. The lipid molecules are amphipathic in nature and are arranged within the membranes by help of two types. Polar hydrophilic end, the form of water loving head which faces towards the outer side of the cell membrane, to interact with the aqueous environment on both sides. Non-polar hydrophobic ends. This region is in the form of water repelling tail, both ends of which face each other, that occurs towards the center of the cell membrane. The portion of lipid molecules varies in the plasma membrane of different cell types. These are forms of cholesterol, 25 to 32%, and mainly of phosphoglycerides. Phospholipids is 55 to 75%. Now look at the protein, how much protein is got. Depends upon the ease of extraction. The erythrocytes, RBCs, has approximately 52% protein and 40% lipids. 52% protein and 40% lipids. Integral proteins. They have stronger proteins and are buried partially or totally in the phospholipids. They are totally the phospholipids. Peripheral proteins. They have weaker association and are bound to lipids of membranes by electrostatic interactions. Carbohydrates. These constitutes about 1 to 5% of the chemical composition. It's only 1 to 5% carbohydrates. Whatever rice we eat is full of carbohydrate, it doesn't go to the RBCs, right? These are associated with the phospholipids or with the peripheral proteins to form glycolipids and glycoprotein respectively. So we have glycolipids and glycoprotein. Glyco is carbohydrate, lipid is fat. Glyco is carbohydrate, protein is protein. The structure of the plasma membrane, various models are given, out of which most accepted model is fluid mosaic model. Remember the name, fluid mosaic model. So what is this fluid mosaic model? The lipid bilayer and integral proteins appear like a mosaic arrangements, and the quasi-fluid nature of lipid enables the lateral movement of proteins within the overall bilayer. This ability of protein to move within the membranes indicates the fluidity of the lipid parts. So look at the fluidity of the membrane. The fluid nature of the membranes is important from the point of view of interactions of molecules within the membrane, as well as other functions like formation of intercellular junctions. It also is helpful in cell growth, secretion, endocytosis, cell division. The membrane occurs mainly by two methods. Active transport is the movement of the molecules across the membranes. Against their concentration, across their concentration gradient. It occurs in few ions and molecules, sodium, potassium pump, sodium and potassium pump. Polar molecules require a carrier protein of the membrane to facilitate their transport across the membranes, because they cannot pass through the non-polar lipids. They cannot pass through the non-polar lipid bilayers. Now look at the passive transport. We have seen the active, now it's the passive transport. Passive transport is mode of movement of molecules or substances across the membrane, without any requirement of energy. Without any requirement of energy. It can be following three types. One is osmosis. It is the process by which water molecules pass through a membrane from a region of higher concentration to a lower concentration. A region of higher concentration to lower concentration. Next is simple diffusion. In this process, neutral molecules move across the membrane along the concentration gradients. Okay. Example, gases and small molecules. Facilitated diffusion. The molecules are transported along concentration gradient by help of ion channel and permeases. Energy is not required in this process. Functions. Cell membrane performs. It is a selectively permeable or semi-permeable membrane. Allows only selected substances to pass inwardly. Next is, it protects the cell from injury. Membranes have carrier proteins for active transport. Cell membranes contain enzymes which perform certain reactions on their surface. They perform the certain reactions on the surface. For example, ATPase, phosphatase, etc. Now, look at the cell wall. It was first discovered by Robert Hooke. Robert Hooke discovered it. It is a rigid and non-living structure which forms an outer covering of the plasma membrane. In plants and fungi. It is present only in plants and fungi, not in any other animals or reptiles. It is absent in animal cells. Okay. Structure of cell wall. Now look at the structure of cell wall. Cell wall is differentiated into three parts. Let us divide them into three parts. Middle lamellae, the layer mainly made up of calcium and magnesium. The layers mainly made up of calcium and magnesium pectacles. It cements the cell walls of two adjoining cells together. It is absent on the outer side of surface cells. Middle lamella along with a cell wall traversed by plasmodesmata, which connects cytoplasm of neighboring cells. Primary cell wall. It is produced inner to the middle lamella. In a young and growing cells. It is capable of growth and extension. It tends to diminish gradually as the cell attains maturity. Secondary cell wall. What is the secondary cell wall? The secondary wall is formed inner towards the membrane to the primary wall. It is towards the inner side. As the cell gets fully matured, its composition is similar to the primary walls. Composition is similar to the primary wall. Functions. Look at what are the functions. Cell walls performs following functions. One, a definite shape to the cell and also protects the protoplasm. It also protects the protoplasm against any mechanical injury. Against any mechanical injuries. Example, damage or any infections. It also helps in cell-to-cell interaction. Cell-to-cell interaction. It provides barriers to undesirable macromolecules and attacks of pathogens. Next slide. It consists of nuclear envelope, endoplasmic reticulum, Golgi complex, lysosomes and vacuoles suspended in the cytoplasm. These are considered together as an endomembrane system because their functions are coordinated with each other. In spite of this, that each membranous organelle is distinct in terms of its structure, and functioning. Okay. Number one, endoplasmic reticulum. Number one is endoplasmic reticulum. The endoplasmic reticulum is a complicated system of membranes. It's a complicated system of membranous channel and flattened vesicles. Membranous channels and flattened vesicles. It is revealed from the electron microscope studies of eukaryotic cells that there is a presence of network or reticulum of tiny tubular structures that are being scattering in the cytoplasm. Endoplasmic reticulum, the endoplasmic reticulum divides the intracellular space into two main components. Luminal, that is inside endoplasmic reticulum compartment, extraluminal, luminal and extraluminal compartment. Types of endoplasmic reticulum. So what are the types of endoplasmic reticulum? Endoplasmic reticulum is mainly of two types, depending upon the nature of its membranes. Smooth endoplasmic reticulum, they do not bear ribosomes in the form of granules on their surface. They act as a major site for the synthesis of lipid and also help in synthesis of steroidal hormone in animal cells. So now look at the rough endoplasmic reticulum. What is a rough endoplasmic reticulum? They are found extensively and continuous with the outer membranes of the nucleus. They have rough membranes because they bear ribosomes being attached to their surfaces. Now look at the functions. It provides support to the colloidal cytoplasmic matrix. Helps in the rapid intracellular transport of the material. Endoplasmic reticulum membranes contain a variety of enzymes for various metabolism. It has a variety of enzymes for various metabolic process, ATPase, phosphatidase, etc. Now look at the Golgi apparatus. Camillo Golgi was the first who founded the Golgi apparatus, so that's the name. Golgi complex or Golgi apparatus is a major complex protoplasmic structure being made up of many flat disc-shaped sacs of cisternae, 0.5 to 1 micrometer in diameter. Occurrence, Golgi complex occurs in all cells except prokaryotes, that is PPLO, bacteria, cyanobacteria, and some eukaryotes. Eukaryotes such as the human red blood corpuscles, sieve tubules of the plant, etc. Cisternae of the Golgi apparatus are found stacked parallel to each other. They are concentrically arranged near the nucleus with two distinct phases. Cis phase forming the phase. This is convex in shape that lies towards the cell membrane and is responsible for receiving secretory materials. Through the transitional vesicles which are pinched off from the smooth endoplasmic reticulum. Trans phase, maturing phase. This is the concave shape that lies towards the nucleus and is responsible for releasing the materials which is being secreted by cis phase and modified in the cisternae. It's modified in the cisternae. So look at what are the functions of this Golgi apparatus. Golgi apparatus perform various functions. It is involved in the formation of lysosomes, vesicles that contains protein and remains within the cells, and they remain within the cell. It performs the function of packaging materials. It performs the functions of packaging materials. It acts as an important site for formation of glycoproteins and glycolipid. It helps in the production of complex carbohydrates other than glycogen and starch. It helps in the formation of cell wall. Lysosomes. These are membranes bounded vesicles. What is lysosomes? These are membranes bounded vesicles that are produced by Golgi apparatus. They are rich in several hydrolytic vegetative enzymes. They are rich in several hydrolytic digestive enzymes, as these are optimally active at the acidic pH, that is less than 7. They are also called acid hydrolase and are capable of digesting macromolecules from various sources like carbohydrates, lipids and nucleic acid. So what are the functions of it? They digest the food contents. They also perform extracellular digestion. The old and useless organelles of the cells. They also have functioning in the cell division. Have functions in the cell divisions. Vacuoles. Next is the vacuoles. Vacuoles are a large membranous sac that is found in the cytoplasm. These stores substances that are not essentially useful for the cell, like water, sap, excretory products and other materials. Okay, they are all stored in the vacuoles. Now look at the plant vacuoles. Plant vacuoles contain not only water, sugar and salts, but also contain pigments and toxic molecules, and also occupy up to 90% of the volume of the cell. 90% of the volume of the cell. The vacuoles is bound by a single membrane structure known as tonoplast. Which in plant cells facilitate the transport of material and some ions against the concentration gradient inside the vacuoles. The concentration of material is tend to be the higher in the vacuoles than to be the cytoplasm. Okay. Animal cells also have vacuoles. They are much more prominent in cases of plant cells. Okay. In animal cells, it's not that prominent. Plant cells have typically large central vacuoles filled with a watery fluid. Following types of vacuoles are being found in different organisms. What are the vacuoles found in the different organisms? Okay. Number one, contractile vacuole. They play an important part in the osmoregulation and excretion. Example is amoeba. Okay. Next is the food vacuoles. What is the food vacuoles? They occur in cells of mainly protozoa and protists. These are formed by engulfing the food materials. They are formed by the engulfing of the food materials. Mitochondria are membrane-bound cell organelles. They are membrane-bound cell organelles essential for aerobic respiration. Okay. Remember the word aerobic respiration of eukaryotic cells. These are also known as powerhouse of the cells. They produce cellular energy in the form of ATP. Occurrence. Mitochondria are present in all living cells except prokaryotic cell and mature RBCs, red blood cells. It is revealed from the studies that mitochondria is not easily visible. Shape and size. Mitochondria vary considerably according to the shape and size. They have varying shapes such as granular fimbrilar, spherical, oval, discoidal, etc. Fimbrilar, spherical, oval, discoidal, etc. Average size of the mitochondria is 2 to 6 micrometer in length and 0.5 micrometer in diameter. Typical cylindrical or sausage-shaped mitochondria has diameters of 0.2 to 1 micrometer. Look at the complete structure of it. A mitochondria contains two membranes. One is the outer and the inner. It's obvious. Outer and the inner. Out of which the outer membrane is smooth and forms the continuous boundary of the organelles. The inner membrane is semi-permeable to some metabolites. It is infolded into the matrix as incomplete partition called cristae. The cristae are responsible for increasing the physiological active area or surface area. The outer and inner membrane divides its lumen into two aqueous components separately. The outer and the inner component. The inner component is also called matrix. The matrix also possesses single circular DNA molecule. A few RNA molecules. Single circular DNA molecule and few RNA molecules. The two membranes of mitochondria have their own specific enzymes associated with mitochondrial function. So what are their functions? Mitochondria provides important intermediates for the synthesis of several biochemicals like pyrimidines, alkaloids and few other things. Okay. The inner chamber matrix of the mitochondria has enzyme for the synthesis of fatty acids. Synthesis of the fatty acids. They help in the regulation of cellular metabolism. Regulation of cellular metabolism. It also helps in the apoptosis. Next slide. These are semi-autonomous organelles that have double membrane envelope. Double membrane envelope. Plastids have their own genetic materials. DNA, due to their large size, they are easily seen under the microscope, normal microscope, not electron microscope, normal microscope. So, plastids are found in all plant cells and are euglenoid except in some protistans, Euglena, Dinophyceae, etc. Types of plastids are differentiated into 3 different types on the basis of the color. Types of pigments found in them. Leucoplast, these are colorless plastids of varied shapes and size. Colorless plastids of varied shape and size with which stored nutrients in the form of carbohydrates, lipids and proteins. Amyloplasts are the carbohydrates. Starch containing leucoplast. Rice, wheat, potato. Okay. Elaioplast are the leucoplast which stores oil and fats. Tuberose endosperm of castor seeds. Endosperms of the castor seeds. Aleuroplast are the protein storing leucoplast. Example, maize, aleuroin cells. Chromoplast. These are the leucoplasts which are yellow or reddish in appearance because of the presence of fat soluble carotenoids, pigment keratin. Chloroplast. These plastids which are greenish in color containing photosynthetic pigments, chlorophylls and carotenoids. So what are the examples? The synthesis of organic food from an inorganic raw material in the presence of sunlight. Okay, that is the example. So now look at the chloroplast. Chloroplast occurs in major number in the photosynthetic mesophyll cells of leaves and green stem. Shape and size. They may be of lens shaped, oval, spherical, discoid or even ribbon like organelles. They also have variable length 5 to 10 millimeter and width 2 to 4 millimeter. Now look at the numbers. This numbers also varies from 1 per cell of Chlamydomonas, a green algae. Chlamydomonas is a green algae to 2 to 40 per cell in mesophils. Ultrastructure. Chloroplasts are also bound by double membrane envelope like mitochondria. The two membranes are smooth and are thick of about 90 to 100 Armstrong. The inner membrane of the chloroplast is less permeable than other ones. The inner membrane is grounded by a space known as stroma or matrix. Thylakoids are a number of membranous like flattened structure that run throughout the matrix or stroma. The several thylakoids are organized in a stacks called grana or integral thylakoids. Many flat membranous tubules interconnect the thylakoids of different granules known as stroma lamellae. Stroma lamellae. So now look at what are the functions in it. It helps in photosynthesis. Formation of organic compounds. Photosynthesis is basically formation of an organic compound. In consumption of carbohydrate and release of oxygen in photosynthesis. Okay. The next may also change in chromoplast in order to provide colors to many flowers and fruits. Gives many colors to many flowers and fruits. Okay. Then helps in storing fat and lipids. Okay. Look at the function in transduction of the energy. Look at the functions. Okay. Next slide. These are small subspherical granular organelles not bound by any membranes. It is the ribosomes. They are the small spherical granular organelles not bound by any membranes. Ribosomes were first absorbed by George Palade as the dense particles under the electron microscope. Hence are also called as Palade particles. P-A-L-A-D, Palade particles. Ribosomes are mainly composed of ribonucleoproteins. Ribonucleoproteins is mainly composed of ribonucleoproteins, that is RNA proteins, RNA plus proteins, and are also known as protein factories as they are primarily involved in the synthesis of protein or polypeptides. Involved in the synthesis of protein or polypeptide. As we have studied earlier, the prokaryotic ribosomes are 70S type, while the eukaryotic ribosomes are 80S type. Both 70S and 80S ribosomes contain two subunits, the smaller and the larger subunits. Let's look at the next slide. The network of interconnected proteinous filaments and tubules which extends from the nucleus to the plasma membrane in eukaryotic cells. What are their functions? Okay, this is cytoskeleton. And so what are the functions of cytoskeleton? The cytoskeletal structure maintains the shape of the cell and its extension. It is also involved in many functions in a cell as a mechanical support motilities. That is what its function is. Okay. Cilia and flagella. These are hair-like projections of cell membrane. Both cilia and flagella are almost identical in structures, but differ somewhat in length. As cilia are small structures working as oars, while flagella are comparatively longer in size than cilia and they are responsible for the movement of cells. Flagella are also present in prokaryotic bacteria, but these are structurally different from that of the eukaryotic flagella. According to in the electron microscope studies, it is predicted that cilium or flagellum are covered with plasma membrane. Their core called axoneme contains a number of microtubules. Running parallel to long axis. The axoneme has 9 pairs of doublets of peripheral microtubules that are radially arranged and a pair of centrally located microtubules. There are a pair of centrally located microtubules. This arrangement of axonal microtubules is referred to as 9 plus 2 array. It is referred to as 9 plus 2 array. The central tubules are connected by bridges and is also enclosed by a central sheath which is connected to one of the tubules of each peripheral doublets by a radial spoke, which is tubules of each peripheral doublets by a radial spoke. The peripheral doublets are also interconnected by linkers. Both the cilium and flagellum emerge through the centrioles like structure called the basal bodies. The structure called basal bodies. The nucleus in the interface as a loose and indistinct network of nucleoprotein fibers called chromatins. The chromosomes are meant for the equal distribution of genetic materials. A single human cell has approximately 2-meter long thread of DNA. 2 meter long, this is about 2 meter. So this finger to this finger when I am stretched out. Distributed among the 46, that is 23 pairs of chromosomes. Chromatin is composed of DNA and some basic proteins called histones. Some non-histone proteins and DNA are also present in the chromatins. They are also present in the chromatin. Each and every chromosome is composed of primary constriction or the centromere. On the sides of which the disc-shaped structure are present known as kinetochores. On the basis of the position of centromere, the chromosomes can be classified into 4 types. So what are they classified into? Number 1 is metacentric. It has chromosome with equal arms and centromere lies in the center. Next is submetacentric. It has one short arm and one long arm with centromere slightly away from the middle of the chromosome. Next is acrocentric. So what is acrocentric? It forms one extremely short and a very long arm of centromere. The centromere is located near the end of the chromosome. So very short loop, right? Next is the telocentric. It has the terminal centromere and it is placed on the extreme end. And telocentric chromosomes are not present in humans. They are not present in humans. Okay? That is telocentric chromosome. Few chromosomes have a non-straining secondary constrictions being present at a constant location at some or other time, which gives the appearance of a small fragments known as satellite. So look at the functions of it. Okay? They control the cellular differentiation. Contains all hereditary information located in the gene. Forms a link between the offspring and the parents. They introduce a variation through the process of crossing over. It controls the cell metabolism. Okay. Moving on to the next slide. These are the microbodies. These are the membrane-bound cytoplasmic elements that are composed of enzymes and other substances These are minute vesicles found in both plant cells and animal cells. Both plant cells and animal cells. Example, in the liver, kidney, protozoa, yeast and many other types of cells, their shapes can be ovoid, spherical, granular. They can be ovoid, spherical or granular. That we almost finished the study of the cells. Okay. Centrosome is the organelle that generally have two cylindrical structure known as centrioles. They are basically surrounded by an amorphous pericentriolar material. Both the centrioles in a centrosome lie perpendicular to each other, in which each has an organization like the cartwheel. They are usually made up of 9 evenly spaced peripheral fibrils, triplet in nature of tubulin protein and tubulin protein. Fibrils are absent in the center. The arrangement is therefore called 9 plus 0. Fibrils run parallel to one another but at an angle of 40 degrees. Each fibril is made up of 3 sub-fibers. Each fiber has a diameter of 20 nanometer. The tubules of the peripheral triplet fibrils known as radial spokes. Let's look at the function of centrosomes. Centrosomes and centrioles perform the functions that are very important. They form spindle fibers and move to the poles at the time of cell division. Which thus help in the movement of chromatids in daughter cells. Next, they help in the formation of cilia and flagella of the cells. Moving on to the next slide, it is specialized and principal organelles of the cell, which contains all the genetic information for controlling at essential processes related to metabolism and hereditary transmission. Nucleus was first described by Robert Brown. The name chromatin was given by Fleming when the material of the nucleus was stained by the basic dye. The dye at that time stained it as a nucleus is known as the largest cell organelle. Largest, remember the word largest. Also known as brain of the cell. Brain of the cell. Occurrence. A nucleus is known to be present in all eukaryotic cells except a few cells like red blood corpuscles of human beings, sieve cells of vascular plant, sieve cells. These are two important. Ultrastructure. The interface nucleus has highly extended and elaborate nucleoprotein fibers called chromatin, nuclear matrix and one or more spherical bodies called nucleoli. Functions. It stores information that controls cellular functions. Next, it controls the synthesis of structural protein. It also stores the genetic information for development, reproduction and the behavior. It also induces genetic variations. Now look at it in the next slide. Okay. The nucleus in the interface has a loose and indistinct network of nucleoprotein fibers called chromatin. The chromosomes are meant for the equal distribution of genetic material. A single human cell has approximately 2 meters long thread of DNA distributed among its 23 pairs. Chromosomes. Chromatin is composed of DNA and some basic proteins called histones. Some non-histone proteins and RNA are also present in the chromatin. Each and every chromosome is composed of a primary constriction or the centromere, on the sides of which the disc-shaped structures are present, known as kinetochores. On the basis of the position of centromere, the chromosomes can be classified into 4 types. There are 4 types we need to remember. One is the metacentric. It has the chromosome with equal arms and centromere lies in the center. Next, submetacentric. It has one shorter arm and one longer arm with centromere slightly away from the middle of the chromosome. Next, acrocentric. It forms one extremely short and one very long arm, and the centromere is located near the end of the chromosomes. Next is the telocentric. It has the terminal centromere, that is, centromere is placed at the extreme end. Telocentric chromosomes are not present in human. Few chromosomes have a non-straining secondary constriction. Being present at a constant location at some or other time, which give the appearance of a small fragment known as satellite. They are called as satellite. So let us look at the function. Control cellular differentiation. Number 1 is controls the cellular differentiation. Number 2, contains all hereditary information located in the gene. Next, forms a link between the offspring and the parents. Introduce variations through the process of crossover. Next, control cell metabolism, central metabolism. Okay, those are the 5 functions. Next, look at the membrane-bound cytoplasmic elements that are composed of enzymes and other substances. They are composed of enzyme and other substances. They are minute vesicles found in both plant cell and the animal cells. They are present in the plant cell and animal cells. In the liver, kidney, protoplaza, yeast and many other types of cells, their shape can be ovoid spherical granules. Thank you.